Hello, everyone. Good morning or afternoon, wherever you happen to be joining us from. This is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to today's webinar, which is, as this series always is presented by the European Society for Toxicology in Vitro and AICCT. This is part of both of our society's efforts to increase and promote computational and in vitro toxicology science and policy, and offer platforms for discussion and learning. Each of our organizations also has annual meetings and offers training and education and support for early career scientists. If you want to learn more about our societies, I'd encourage you to go to our websites. So just brief housekeeping for the webinar. The webinar is recorded, and the recording will be posted on the AICCT webinar page a few days from now. Presenter slides will not be available this time, but you will be able to see the recording. Also, the lines are going to be muted. If you have a question, I would encourage you to enter it into the Q&A box at the bottom of your screen, the toolbar there. You can just click on Q&A and enter your question. You should be able to see other people's questions. If you have a similar question, you can vote it up with a thumbs up so that it goes up higher in the priority field. So we'll collect your questions during the talk, and then at the end of the presentation, we will ask them of the presenter. You can also, if you have technical questions, you can reach us in the chat. So just a brief mention of a couple of upcoming activities. The ESTIV Congress is again in Barcelona next year. So I think things will be starting to get moving on that conference soon in terms of programs and abstract submission and all that. So that's going to be exciting. ESTIV is also holding an applied toxicology course in October, and so I would encourage you to find out more about that if you'd like to attend. And then finally, I'm very happy to announce that AICCT's 10th annual meeting is open for abstract submission. We have a number of oral presentation themes, which you can see on your slide there. It's going to be October 12th through the 14th. It's a virtual meeting. The abstract deadline is July 15th. So if you want to apply to give a talk or present a poster, please visit the website to find out more about that. We have a really exciting lineup of speakers to be anchoring these session themes. So I'm really looking forward to it. And we hope you will submit your work and participate. There will also be student and early career scientist awards and other engagement opportunities, and panel discussion, or maybe even a debate. So if you want to visit our website again, sign up to receive our emails, then you will learn when more information becomes available. Now, I'm very pleased to announce today's webinar. It's presented by Giel Hendriks of Toxys. Dr. Hendriks has a PhD in molecular biology from Utrecht University and has worked for 4 years as a postdoctoral fellow at Leiden University, studying the relationship between DNA damage and gene mutations. After this, he worked as a senior scientist at Leiden University Medical Center on the development of in vitro reporter systems to detect and understand the mechanisms of genotoxicity. So during this period, he was instrumental in the development of the ToxTracker assay. And then in 2014, Giel obtained financing to start Toxys, the company, and bring ToxTracker assay to the market. And now as CEO of Toxys, he worked to develop the company into an internationally recognized contract research organization working to provide assays for genetic toxicology and chemical safety testing for the pharmaceutical, cosmetics, and chemical industry. So without further ado, I'm going to give the floor over to Giel.
Thank you, Christy. Thanks for the introduction. And of course, also a big thanks to STIF and the ASCCT for the invitation to give this webinar. It's really a pleasure. And let's get started, because I have a few things I would like to share with you. Maybe very briefly, although Christy already introduced it, very briefly about Toxys, for those of you who are not familiar with the company. So Toxys is a Dutch biotech company that is developing in vitro assays for toxicological research. But we also offer the assays that we develop as a service. So we're also working as a contract research laboratory. And we have our prime knowledge and expertise in the areas of genetic toxicology, developmental toxicity, and mechanistic toxicity testing. And I will show you a few examples of that today. We are trying to help partners from the pharma, chemical, cosmetics, and food industries. Although we are still a small company, I think we try to be active in various scientific expert committees to maintain that high knowledge. So I don't think I will bring any news to say that human safety testing is crucial for all new pharmaceuticals, chemicals and agrochemical products, cosmetics ingredients, consumer goods, food-related substances. Basically everything that is brought to the market needs to be thoroughly tested for safety to humans. But not only to humans, but also safety to the environment. So ecotoxicity is definitely becoming a more and more important field. And as well as occupational toxicity. So toxicity to workers that are actually working in the factories, in the plants that are developing these new or producing those new products. And in many areas, we see that the assays that we use for that safety testing, they don't always perform very well. So there's either a low sensitivity, meaning that the assays that we are using are not always predicting the potential hazards for humans. But maybe a bigger problem is the low specificity. So that's a relatively high number of false or misleading positives, in vitro positive tests that do not reflect what's happening in vivo or in humans. And partly this because many of the, let's call it old school tests or approaches often fail to produce insight into the mode of action of a toxic substance. And then it makes it very difficult to translate information from your in vitro assay to humans. Consequences, well, for the industry, that means unnecessary investment in hazardous compounds and the loss of potentially very valuable lead compounds. But I think also importantly, needless animal testing, and which are expensive, time consuming. Obviously, the animals are not in favor of animal testing, and so there's definitely also some ethical questions. However, animal testing is still, I think, required for most of the regulatory applications. And last year, the European Commission published a report that kind of outlined the use of animal testing. And it was clear that still a large number of animals are used, over 10 million. And the areas that really stand out are reproductive and developmental toxicity that cover almost 50% of all animals used, but also areas like repeated dose testing, but also carcinogenicity and genotoxic testing are high on that list. So basically the conclusion is that there's a clear need for in vitro assays, or at least novel approaches to better understand the toxicity of compounds and to better predict the human risks. Now, an approach that many laboratories, many research groups, and also companies, including we at Toxys, used is, and this was basically started about 10 to 15 years ago when toxicogenomics and whole genome transcription profiling became fashionable. Was started that it was very simple. The reasoning is that when chemicals interact with molecules in cells, in organs, in tissues, they might inflict damage to those structures, And this will be recognized in the cells by all kind of sensors that will activate all kind of signaling pathways to repair the damage or signal to other cells that there is stress to activate the immune system. And those stress pathways will activate gene expression of all kind of proteins. And these can be very common stress pathways, but also very specific pathways that are activated in relation to the damage that is induced. And the idea was that if we can capture that whole change in transcription of the entire genome or transcriptome, then change in gene expression might be giving an indication on the mode of action and on the toxicity of compounds. And so this is an approach that also, when I was still working at the university a long time ago, that we took. So we started using mouse or human cells, expose these cells to all kind of toxic substances, and look for changes in gene expression. So doing whole genome transcription profiling, identify a biomarker panels, and use that to identify key player in those responses. And then in that area, we try to use those genes, those biomarker genes, as a reporter. So we often, either by antibodies or by fusing fluorescent proteins to it, use them as an indicator for identifying toxic responses related to exposure. Now, today I would like to show you 3 examples of assays where we have used at Toxys that approach. And I will first focus on genetic toxicology and an assay that we call ToxTracker, really aim to identify the first of all genotoxic substances, but also understand the mode of action of these substances to better predict the effects that we will anticipate in vivo or in humans. An assay that I will not cover today because of time limits is an assay that we have named ReproTracker. So this is a human IPS, a human stem cell based biomarker assay to identify developmental toxicity. But I will not cover that. And a second assay that I would like to cover is an assay called Tox Profiler, which again using signaling pathways and biomarker activation to get a broad toxicity profile of substances, again focused on understanding the mode of action. But I would first like to go to the area of genetic toxicology. So this strategy for gene tox testing is well defined in the different regulatory documents that are around, issued by the different international organizations and set in OECD guidelines. Basically, it's a combination of first in vitro testing, where novel chemicals, pharmaceuticals, food ingredients, cosmetics are tested for the ability to induce mutations, as well as chromosomal aberrations, and occasionally also chromosome number changes, so aneuploidy. Typically, this consists of the Ames test and the micronucleus assay. And then follow up in vivo testing, again addressing the same endpoints: mutagenicity, chromosomal aberration or clastogenic, and aneuploidy. In the transgenic rodent assay, nowadays also big A assay for mutations, and mouse lymphoma, micronucleus assay in blood or bone marrow, and occasionally also the in vivo COMET assay. But obviously, before going into the more regulatory assays, it makes a lot of sense to start off with a thorough screening using in silico approaches, QSAR models, as well as in vitro screening assays, to in a very early stage already identify genotoxic properties of materials. And this screening typically consists of in silico models, like the Derek and Sarah Nexus suite from LASA or LeadScope GeneTox suite. Miniaturized versions of the regulatory assays, like a mini version of the Ames test, micronucleus assay by flow, COMET chip. But over the last decade or so, there have also been a number of newer approaches where cellular signaling biomarker assays have been developed. This is obviously where ToxTracker fits in, but also examples like the multi-flow assay by Litron Laboratories in the US, or toxicogenomics approaches by the TGX-TDI platform initially developed at Health Canada. And these can really provide a lot of mechanistic insight into the genotoxic properties of these compounds. ToxTracker is a stem cell based reporter assay using mouse stem cells for the reason that these are untransformed cells, have a normal cell cycle control, have a normal DNA damage response. So they are not cancer cells. And in these cells, we've integrated fluorescent reporters, and each reporter is activated upon a certain type of cellular stress, and in this case related to the induction of DNA damage, as well as to oxidative stress and protein damage that can indirectly cause genotoxicity. And together, combined in one assay, ToxTracker can provide insight into the mode of action of genotoxic compounds. This assay is typically used on one end as a kind of a high throughput screening platform for novel substances, but obviously also as a more follow-up assay to really dive into the mode of action of genotoxic substances and to understand whether a certain result is actually relevant for in vivo or for human exposure. Now, I said the biomarkers, those 6 biomarkers that are included in ToxTracker, they came from toxicogenomic studies. I won't go into detail too much, but to give you an idea, on the left you see the 2 biomarkers in the assay that detect the induction of DNA damage. First gene, and I will go into a little bit more detail on the next slide, but gene called BSCL2 that detects the formation of bulky DNA adducts. And a second biomarker, RTKN, is triggered by the formation of DNA double-strand breaks. Then third marker, BTG2, detects activation of the p53 response, the tumor suppressor response, strongly associated with genotoxicity, but can also be triggered by other toxicological events. There's 2 markers for the oxidative stress induction that can indirectly cause genotoxicity, 2 of the major antioxidant responses in the cell. And finally, there's a marker for DDIT3 for the protein damage stress response. When we identify these biomarkers, obviously the genes were unfamiliar. They were poorly classified in literature. And so we needed to perform a little bit more testing to understand what it means if one of those biomarkers is activated. And I will give you one example, but we have done this for all of these genes. So the first gene that came out of those toxicogenomic study and was activated by DNA damage is a gene called BSCL2. It is associated with a hereditary disease that's related to problems in the differentiation of fat cells, adipocytes, and also to neural diseases. But surprisingly, the gene was very strongly activated by DNA damage. So as you can see here in the picture, upon exposure to cisplatin, the DNA crosslinker, this gene is definitely activated. And because we have these fluorescent reporters built into those cells, we can actually follow this induction in time, giving a kinetic and also a dose response impression of how the cells respond. And you can nicely see that the cells become green upon exposure to cisplatin. Obviously, we had no idea what the function of this gene was. And then, so we reasoned that at least one of the triggers for a genotoxic response would be problems in DNA replication. Because this is where DNA damage inhibits the DNA replication fork. And so this is what we studied. And first of all, we confirmed that indeed it was inhibition of DNA replication that activated this gene by exposing the cells to 2 DNA replication inhibitors, acetylcholine or hydroxyurea. So without inducing any DNA damage, still in blue, you see that this gene is activated, while a second reporter for oxidative stress was hardly affected. And then we followed up by using small molecule inhibitors of various kinases in that whole signaling pathway. And we were able to confirm that indeed when we added inhibitors for the ATR or CHEK1 kinase, so basically blocking the right part of that signaling cascade, that if we inhibited that, then we lost that activation of that BSCL2 gene. So confirming that indeed it's replication stress and the kind of classical signaling pathway that is triggering activation of this gene. And so by that way, we could really understand what was going on when this gene was activated. Now, what does it then actually look like? So ToxTracker is performed in 96 or 384 well plates, and activation of those fluorescent reporters is detected by flow cytometry. Makes it very quantitative. You can measure basically on a single cell level. And you can also simultaneously look at cell survival by measuring, let's say, the fraction of surviving cells. Here on the screen, you see 4 examples of 4 genotoxic agents. Cisplatin is a DNA cross-linker. And here you see that with an increasing concentration in red, the 2 DNA damage reporters are activated, indicating to us that we get formation of bulky DNA adducts. And we would predict that because of that, the compound is likely mutagenic, and it would, for instance, be positive in an Ames test. We also see activation of that second DNA damage reporter, meaning that there's a formation of DNA double-strand break. So it's the clastogenic effect, there's chromosomal aberrations. In gray, there's activation of p53, very little oxidative stress or protein damage. The dashed line, horizontal dashed line, indicates a 2-fold increase in GFP signals. That is a threshold for a positive test result. Etoposide is a topoisomerase poison that traps the topoisomerase complex, or basically traps the replication fork at the DNA. Also there we activate both of the DNA damage reporters. Third example, methylmethane sulfonate, a methylating agent, methylate DNA. So we activate again in red both of the DNA damage reporters. There's in gray p53 activation again. But now we also see that there's quite a lot of oxidative stress. And this is well described because MMS also methylates proteins. You get depletion of glutathione and basically loss of your cellular antioxidant system. A different profile was obtained for a tubulin poison, Taxol, so that disrupts the microtubules, traps the cells in mitosis. There's no inhibition of DNA replication because there is no activation of the BSCL2 reporter, but there is a strong activation of that second DNA damage reporter, showing already that the different mechanism of action also is reflected by different responses in this system. We expose the cells to compounds that induce oxidative stress, diethyl maleate or sodium arsenide. We see a lot of oxidative stress reflected by the blue lines, the oxidative stress reporters that are activated. And in gray, there's also p53 activation again, confirming that not only DNA damage, but also oxidative stress can activate the p53 response. Arsenide also reacts with protein. So in green, we now also see activation of that protein damage reporter. But if we expose the cells to compound tunicamycin that inhibits folding of nascent protein chains in the endoplasmic reticulum, or nitrophenol that basically denatures proteins, we selectively see activation of that protein damage reporter. So really emphasizing the specificity of those responses. Sometimes compounds require metabolization in the liver, and that is not a feature that is present in those ToxTracker stem cells, but can be added by the addition of red S9 liver extracts, arylchlor, but nowadays also phenobarbital in juiced rats. We've optimized the protocol a bit for this system, but the bottom line is that in the absence on the left of S9, a compound like aflatoxin B1 is negative. There's no toxicity. There's no activation of any of the reporters. But once you add S9, the compound becomes metabolized and becomes also very genotoxic. So again, both DNA damage reporters are activated. There's p53 activation. There's toxicity observed. So the system is fully compatible with an S9 metabolizing system. Obviously, to make sense of all those data, once you start testing more compounds, data increases rapidly. We have generated a software tool that we call ToxPlot to analyze those flow cytometry information. It basically calculates a level of reporter activation as a function of toxicity. It also allows us at equitoxic doses to compare the responses for different compounds. But more recently, we've also added the ability to really do the BMD quantitative dose response modeling, really use the data to fully analyze the dose response and calculate those BMD benchmark dose responses. So a typical readout for the assay from the software is that activation of those biomarkers are reflected in this heat map, where activation of the gene is of the reporter is indicated by green, and the darker the green, the stronger the induction. And you can immediately see that in red, those DNA damaging agents primarily activate the DNA damage reporters and p53, while in blue, those oxidizing agents primarily activate the oxidative stress reporters. But it's good to realize that behind these relatively simple heat maps, there's a lot of quantitative information on activation of those reporters. where at different levels of toxicity, in this case 10, 25, and 50%, you really get a quantitative measurement for activation of those different responses, really allowing to look for primary and secondary effects in a dose-dependent fashion. But as said, a more quantitative measure you can get from applying the benchmark dose approach, where you can basically model the dose responses through the measurements and calculate a range of concentrations that is able to activate to give a certain benchmark response or a critical effect, as is also often referred to. And in collaboration with Paul White at Health Canada, we've done a lot of work in using the information ToxTracker to really calculate those BMD approaches, which you can use to rank compounds, to look for potency. And it's very useful if you look for potency of different, let's say, structure analogs or different compounds in a certain area. And that worked really nicely. So we could really use that BMD approach to rank, in this example, over 120 compounds based on their potency to induce DNA damage. Where on the left, the top left, those are the most potent compounds, where in the bottom right, these are compounds that do not or hardly induce any genotoxic effect. The BMD approach can also be useful when you want to extrapolate the information from this in vitro assay to in vivo. So this was a nice collaboration with John Wales and George Johnson, UK, Cambridge University and Swansea University, where we compared the BMD concentration from ToxTracker with the BMD values from the in vivo micronucleus assay. And it turned out that the correlation was very, very good. So potent compounds in ToxTracker were also the compounds that were potent in inducing micronuclei in vivo, confirming basically the high sensitivity and specificity of the ToxTracker system to identify in vivo genotoxins. And that is also reflected on this slide, where over the last years, we've tested really quite a large number of substances in this ToxTracker system. And then when you do the calculation, how well the assay is able to identify genotoxic compounds, sensitivity and specificity is well over 90%. So really doing a good job, and actually a better job than the individual regulatory assays that we are currently using. Obviously, if you combine those regulatory assays, the sensitivity improves significantly, but specificity remains a challenge. And to illustrate that, I would like to show you two comparisons. First of all, we did a comparison with activation of that BSCL2 marker in ToxTracker and said, so how well does it now predict the outcome from the, let's say, standard regulatory assays? And we combined information from the Ames test and the mammalian mouse lymphoma assay. If we did a comparison on every assay itself, it would be about 70%. But if you combine Ames and mouse lymphoma, then the correlation with the ToxTracker assay is very good. So if the BSCL2 gene is activated, then in nearly all of the cases, that compound is also positive in the Ames or mouse lymphoma assay. And the negative was also confirmed in Ames and mouse lymphoma assay. Now, we did the second comparison with the RTKN marker, the marker for DNA double-strand breaks, and compared it initially to the in vitro micronucleus and chromosomal aberration assays. Again, data were combined, and then positive in ToxTracker was also positive in the in vitro micronucleus assay, but negative in ToxTracker. About 40% of the compounds were negative in the in vitro micronucleus or chromosomal aberration assay, meaning that either ToxTracker was missing part of those genotoxic compounds, or alternatively, the in vitro micronucleus assay was a little bit oversensitive and it was incorrectly classifying genotoxic compounds as genotoxic. And we believe that it's actually the case because, again, in line with the study that I just showed you on the BMD analysis, when we do the comparison between activation of that RTKN marker and the in vivo micronucleus assay, that correlation is well over 90%. And with that, we started investigating whether then ToxTracker would also have its application in a regulatory space. And that ultimately led to the start of an international OECD interlaboratory validation, where 7 laboratories have set up the assay in-house and contributed to an official OECD validation. That's experimentally that the study has been completed, and we're now analyzing the data. But so far, at least the transferability and the reproducibility seems to be very good. I have to say I have not seen let's say the actual data, because Toxys is not actively involved in the study because of conflict of interest. But hopefully we'll be able to publish the result later this year. ToxTracker was already designed to identify the mode of action of genotoxic compounds, but over the last couple of years, we have expanded its applicability and see can we cover more mechanisms of action that are relevant for genotoxicity, because this is actually the information that is lacking from the standard approach that we're now using. And we also believe that this is the information that you need if you want to reduce animal testing and make a better prediction for human safety based on in vitro assays. And I would like to show you 3 examples. ToxTracker Ace, that is really focused on the identification of aneugenic compounds, so compounds that give a change in chromosome number, but are not DNA reactive. Tubulin checker, which is a follow-up of ToxTracker Ace to understand the mode of action of those aneugenic compounds. And ToxTracker AO for antioxidant to investigate the relationship between oxidative stress and potential indirect genotoxicity. For ToxTracker Ace, we've expanded the panel of reporters a bit, and now also included the cell line to investigate cell cycle progression of the cells while they're being exposed, and doing a staining for DNA content, so look for the number of chromosomes in the cells. And that has an important impact. So when we test, in this example, 4 genotoxic DNA reactive substances in the ToxTracker Ace protocol, on the left you see the 4 compounds that all activate both of the DNA damage reporters in ToxTracker, again suggesting to us that these compounds are likely mutagenic, they are DNA reactive, they inhibit DNA replication, and are likely to cause chromosomal aberrations. Now, in ToxTracker Ace, also information on the cell cycle distribution of the cells is included, meaning that in the central part, in the blue peaks, basically show the distribution of the cells over G1, S, and G2 M phase. But you see after 4 hours, there is no effect on exposure to cisplatin, so there's no change in cell cycle distribution. But after 24 hours, clearly cells accumulate in S phase or G2 phase, as expected. But on the bottom right, you can see that there's no change in DNA content, so there's no induction of polyploidy. But if we compare this to 4 compounds that are clearly aneugenic, and so these are 4 tubulin poisons, first of all, you see that basically in most of the cases, only one of the 2 DNA damage reporters is activated. This is the marker for DNA strand breaks, but there's no DNA replication inhibition. Only at the very high cytotoxic doses, we start to see also the second marker being activated. But the biggest difference is in the central part, where after 4 hours, you see that cells accumulate in mitosis. These cells also become phospho-H3 positive. Under the microscope, you can see that there is mitotic arrest, really different from those clastogenic agents. And after 24 hours on the right, cells remain stuck in G2 M. And now also we see the appearance of aneuploid or polyploid cells, so really cells with abnormal DNA content. If this is something that we observe, then of course, one way to follow up is to see if can we further understand the molecular initiating event. And so we've designed an additional reporter cell line where alpha tubulin is tagged with GFP, expressed from this endogenous promoter, so that makes it possible to investigate the tubulin dynamics in the context of a living cell under the microscope or by flow cytometry. Flow cytometry, it also allows us again to look for the cell cycle distribution of the cells. And we can really investigate whether tubulin is the target for that aneugenic compound. I don't have time to go into details, but the long story short, you see that tubulin stabilizers typically give a strong increase in GFP signals, where tubulin destabilizers show a decrease in tubulin signals in these cells. Interestingly, if we add compounds that inhibit cells in mitosis, in this case 3 aurora kinase inhibitors, which block cells in mitosis, there is no effect on tubulin signals, as well as for a number of genotoxic and non-genotoxic compounds. The final extension for ToxTracker I would like to share is ToxTracker AO for antioxidant. And in this case, we use the basic reporters that are in ToxTracker, but now expose the cells in the presence of two ROS scavengers, so compounds that bind to the reactive oxygen species, typically N-acetyl and cysteine and reduced glutathione. And then when we do that, for two examples, on the left, MMS, that this picture I already showed you, and potassium bromate, you see that for MMS in the central part, there's oxidative stress and DNA damage being induced. If we add, in this case, N-acetylcysteine, it almost completely abolishes the oxidative stress response in the cell because basically those oxygen radicals have been scavenged by N-acetylcysteine, but there's no effect on the DNA damage response, suggesting that MMS directly binds to the DNA. And in contrast, if we expose cells to potassium bromate, again, oxidative stress and DNA damage being induced. N-acetylcysteine reduces the oxidative stress, but in this case, that also leads to a complete loss of the DNA damage response, suggesting that genotoxicity for potassium bromate is secondary and related to oxidative stress. So then I would like to switch gears and show you a second assay that is using more or less the same technology that was used for ToxTracker, but in a different area. And this is an assay that we call ToxProfiler. And ToxProfiler was is actually built on a panel of HepG2 reporter cell lines that are built with the same technology that was used in ToxTracker, but was developed within the European flagship program EU ToxRisk, where academia, research institutes, as well as industry are coming together to improve in vitro and in vitro safety testing and move away from black box animal testing. And these HepG2 reporter cells were developed here in the Netherlands at Leiden University by the laboratory of Professor Bob van der Water, who is also the director of the EU ToxRisk program. So ToxProfiler is a panel of HepG2 human liver cells, where again, different biomarkers are included in these cells, stably expressed from their endogenous promoter to capture a wide range of toxicological responses, not only cell death and activation of cell proliferation, so mitogenicity, but also inflammation, protein stress, ion stress, so metals released in the cells, autophagy, problems with the endoplasmic reticulum, also genetic stress here. It looks at the p53 response, which is not exclusively related to DNA damage, as we have seen for ToxTracker, and also at oxidative stress. And this assay is really aimed to give a broad toxicity profile of compounds. Here you see the 7 biomarkers. Again, they came from large toxicogenomic screens and the toxicogenomics databases that are publicly available. And for those different toxicological endpoints, we selected a biomarker that had a key role in that cellular signaling response. And those biomarkers were then again attached to a fluorescent reporter and stably integrated in those cells. So how does the assay work? Well, in the first step, we look for toxicity in those cells to select the dose that we should use for actual testing, and that is based on cellular uptake of propylium iodide in the cells, which indicates leakage of the cells, and in this example, cadmium chloride, you can see that at a certain concentration, cells start to take up propylium iodide, which indicates toxicity. And we typically limit measuring in this system to doses that are non-cytotoxic or at least do not exceed the 15% cytotoxicity level. But obviously, many compounds do not induce any toxicity, and then we stick to the highest dose. Which is typically 1 millimolar for this assay. In this dose finding, we can also look for autofluorescence, something that we also do for ToxTracker. Some compounds that we test are autofluorescent by themselves. And so we during this dose finding, we can already look for autofluorescent compounds. In this case, curcumin, where on the bottom right you can see that at certain dose they become very fluorescent, very bright under the microscope. But obviously, many compounds like acetaminophen, they don't have any fluorescent issues. So in the next, on using high content microscopy, we can look for activation of those different fluorescent biomarkers. And the advantage of using imaging in this case is really that it allows us, first of all, to look for induction of fluorescence, but it also helps us to look for a specific localization of those activated proteins, which makes it so either compounds that accumulate in the nucleus or are migrating from the nucleus to the cytosol. Or for the autophagy marker LC3 localization in those in foci, and this not only increases the sensitivity of the system, but it also allows for different kind of biomarkers to be used. So not only biomarkers that are transcriptionally activated, but also biomarkers that are phosphorylated, that are changing in localization, that are stabilized or destabilized, and but by analyzing the responses in those cells, it is clear that exposure results in selective activation of some of those biomarkers, where, for instance, on top, aneugrophyllite really induces a lot of oxidative stress, here activating SR sulfur reduction 1, and also induces some protein folding problems. Cadmium chloride very strongly activates that protein folding reporter. Mitomycin C is a DNA damaging agent, selectively activates the p52 response, and verapamil strongly activates the autophagy marker. So the selectivity, like for ToxTracker, also for ToxProfiler is there. But obviously, using this imaging, we can get these clear dose response curves that not only allow us to look for the activation of the different cellular responses and understand what is now the primary toxic response that we see, but it's also in relation to the exposure level, where often say different cellular responses or different toxicological responses are activated depending on the exposure conditions and exposure level. For ToxProfiler, we use the POD approach, so the point of departure approach, to quantify the responses and determine what is now the concentration of the dose where we start seeing a certain toxicological response. POD is determined as 2 times the standard deviation over vehicle controls. And that really helps us to quantify the different toxicological responses that are activated, and getting quantitative information on the primary mode of action. So again, here on the large table on the center right, you can see that as an example, on the top CDEO, primary activity in red is inducing oxidative stress because it activates the SRX1 reporter. But with a lower potency, so the higher concentration, it also has the tendency to induce some of the other reporters. Where we see, for instance, for cisplatin, that it primarily activates the P21 response, lowest POD, but at a higher dose, it also activates some of the other reporters. So this really, in a quantitative measurement or in a quantitative way, gives an indication what is now the primary reactivity and what other toxicological responses do we see when we increase exposure. Again, like for ToxTracker, also for ToxProfiler, we generate a little bit easier to interpret heat maps, where typically in this case, the darker the red, the stronger the induction over in this case, a course of increasing doses. And you can nicely see that. And we also cluster compounds with a similar mode of action or a similar toxicity profile together. And it nice, as you can see, it nicely groups together a compound with a similar mode of action. So in blue, a number of those oxidizing agents. In green, for instance, these 3 DNA damaging agents. On the bottom, there's obviously a few compounds that do not show any toxicity. The 2 metals, cadmium and copper, show a similar pattern. So it really helps to understand the mode of action, group compounds together, and really understand the mechanism of action of a toxic substance. the advantage of using the GFP tech biomarkers stably integrated in those cells is that you do not have to manipulate the cells by adding antibodies, but you can really look in living cells, and that clearly has some advantages. So where in the standard tox profiler assay we tend to stick to one time point, the system really has the ability to look for the cellular responses, not only in relation to the dose, but also in relation to the time. And that kinetic information is really useful to understand what's going on, because Tox Profiler typically captures activation of a cellular response. And then the immediate question comes up, is this an adaptive or an adverse response? And information on the kinetics really can be valuable there. So in these examples, we've combined 4 different biomarkers, so not only the sulfur reduction 1 reported as in the standard Tox Profiler panel, but also other biomarkers, biomarker cell lines. Yeah, we have that kind of, they basically describe the entire path between the induction of oxidative stress, detection by the cellular sensors in the cell, activation of gene expression, and expression of the antioxidant system. And by following that in time, on the bottom left, you can really see that it gives an indication whether responses are adaptive, so you see a peak and then the cells recover, or whether there's an adverse response where there's no recovery and basically toxicity increases, and also the sickening increases, so the cells do not recover. So by using that kinetic information, there's additional insight into, let's say, the responses that are captured in this system. Now, a final example that I would like to share is, because the Tox Profiler system is using HepG2 cells, which are liver cells, already, and this is work that has been done within the EU Tox Risk program. An obvious question was, of course, so how well can then the system detect liver toxicity? And a number of these studies have been published, so I can only highlight a few findings. But initially, the selection of compounds was tested according to their DILI, so drug-induced liver injury label that was given by the FDA. It was compared to effects that were observed in the Tox Profiler reporter cells. In this example, SRXN1, and it was actually remarkable to see that that activation of those cellular responses and those fluorescent reporters actually correlated pretty well with the DILI labeling that is known for these substances. And that prediction further increased when also inflammation, which I think plays a crucial role often in liver toxicity. When information on inflammation was included, so by also triggering the cells with TNF alpha, and that validation was extended in 2 more studies. In this example, more than 100 DILI compounds. Compounds were tested, ranked, classified based on their ability to activate those Tox Profiler reporters and compare to their DILI label. And that turned out to actually work really well. So many of the non-toxic compounds or non-DILI compounds also tested negative in the Tox Profiler panel, where compounds with a high DILI label in most of the cases triggered 1 or more of those Tox Profiler responses, with the additional benefit that in those positive compounds, it was clear that there were different mechanisms of action that were actually causing the toxicity of these compounds. So there was also understanding on the underlying mode of action. These studies have been published, so they are available. If you are interested, you can also contact me so we can share them. So to wrap up and to conclude, so I hope that I've been able to show you that both ToxTracker and Tox Profiler are innovative mammalian or human reporter assay that can help to unravel the mode of action of compounds. ToxTracker really reliably predicts in vivo genotoxicity in a quantitative way, especially by using those benchmark response approaches, getting a quantitative measurement and predict effects in vivo and in humans. Tox profile really provides an extensive toxicological profile that can be used to understand the mode of action. Interestingly, it also gives a good correlation with DILI. And those quantitative dose response modeling, either using a point of departure approach or a B&B approach, that can be really valuable for extrapolating data from in vitro to in vivo. The typical application domain that we see for these types of assays is on one hand, of course, very early mechanistic toxicity screening, but also investigations in the mode of action of toxic compounds. And of course, it fits very well in AOP approaches, understanding the pathways from the molecular initiating event to the different key events that lead to an adverse outcome. So AOP and weight of evidence approaches, I think that this mode of action information is crucial. The quantitative information also helps for potency ranking if you're testing multiple compounds in a certain chemical family, or for chemical read across to show that compounds with a similar structure also have a similar behavior in the biological system. And obviously, these assays fit very well in the next generation risk assessment strategies and approaches. And with that, I would like to thank you very much for your attention, and I hope there's still a bit of time for questions.
Well, there definitely is, and we have a lot of great questions. So let me start asking them. Thanks for your presentation. It was a really nice overview of these assays. Okay, so first question is why use, and I think this is relevant to the ToxTracker, but why use mouse stem cell and not human stem cells? Yeah,
that's a question that I get more often. So, but a very good and very relevant question. Well, the honest answer is initially when we started the whole biomarker discovery, the toxicogenomics, for a reason that we wanted to do this in non-cancer cell lines. We wanted to use, let's say, untransformed cells to get an indication on the cellular responses to these, in that case, genotoxic compounds. And obviously we could use primary cells for that. We actually did in those studies also primary liver cells. But if you want to establish a biomarker system later on, you need a stable cells system, and primary cells, they don't really divide, so you cannot really use them as a reporter system. And so stem cells are were in our view the way forward. And at that time, we didn't want to use human embryonic stem cells for all kind of let's say ethical reasons and also regulatory limitations. Human iPS cells were not invented in those days. So mouse ES cells were an obvious choice, and obviously these are widely used also for mutation assays, and so they're genetically very stable. So there was a lot of knowledge. Of course, in time, those iPS cells were discovered, and we had a lot of discussion whether we should actually switch to human iPS cells, and but we decided not to do that for two reasons. First of all, the ToxTracker assay, as it is right now, and has been extensively validated over more than 10 years. It predicts so in vivo genotoxins and human genotoxin with an accuracy of about 95%. So there is very little to gain switching to human cells apart from metabolization. That would be different between mouse and human cells, but both mouse and human cells do not have any metabolizing capacity anyway. And it's also simply not possible to transfer those biomarker genes from mouse to human. Some of those biomarkers are really specific for stem cells, some are specific for mouse. They reflect a response that is also also present, of course, in human cells. But the key players, the key proteins, are somewhat different. So it would basically be going back to the drawing board and start all over. And I said with an accuracy of about 95% detecting in vivo human genotoxic, there's very little to gain. And then finally, to kind of as an advocate of the devil, we are still in the era where the Salmonella Ames test, of course, is the golden standard. So while I do, of course, recognize that the use of human cells is definitely preferred, and we do it for repro tracker, and we do it for tox profiler. I think from where we are right now, ToxTracker is really a big step forward. Hope that answered the question.
I think it's definitely. Okay, there's a question about whether the software, so the ToxTracker and profiler software, are freely available.
They are not. So yeah, no, they're not freely available, and that in a way that that they're tied very much to ToxTracker. So for people that use ToxTracker, this is also available. It's a cloud-based software solution. So you can. So if you use ToxTracker, you can also get access to that software. It runs in the cloud, so it's basically login. It's hosted in Google, but it contains a database of all your tests results. So it means, and that's strictly related to you. So it's dedicated, and that's why we also have to host the software. So it's not freely available, but it's definitely available to everyone who's using ToxTracker.
Great. Another ToxTracker question: how do you rule out a false increase of the signal due to cytotoxicity and not genotoxicity?
Yeah, so it's a fair question. So I have to say that because we use the flow cytometer and analyze the signals in intact, so in living cells, we see that the assay is quite insensitive to cytotoxicity. So it means that even under relatively high cytotoxic concentration, you still get a very, let's say, a robust signal, and also a good relationship to the dose. There is a cutoff where we need at least 25% surviving cells. Mm-hmm. If viability goes below, then we discard the results. But this has been extensively tested, and so there's clear cutoffs of what doses not to include. But because we analyze fluorescence on single cell level in intact cells, we typically get a very nice dose response curve without being too much affected by cytotoxicity.
So there are two questions about the metabolic capability of HepG2 and wondering if you've considered HEPA-RG.
Yes, considered absolutely. Obviously so we obtained those cell lines from the EU Tox Risk, and within that in that program, although BioPredict was involved, all those reports were generated in HepG2 cells. I think also partially because of historical reasons. I don't think at this stage there's no real plans to basically start the development of all those cell lines again using HEPA-RG. I think metabolization HEPA-RG is definitely a little bit more active than in HepG2. I have understood from the scientists within the EU Tox Risk program that they did do some toxicogenomics on HEPA-RG as well as HepG2, and that HepG2 on the transcriptome actually resembles primary hepatocytes more than HEPA-RG, but that is of course not related to metabolization. We do grow these cells in when metabolization is important. We can grow the cells in 3D structures, and then you see that the metabolic capacity of those HepG2 cells also tremendously increases, and they become really much more metabolically competent. Of course, there's also some downsides that it takes longer to mature them, and HepG2 is definitely not completely negative for metabolization. So at this stage, yeah, we've considered it, but at this stage, it would just be too problematic to transfer and build in again all those reporters, which are really artificial chromosomes that are expressed from endogenous promoters. So it is not an easy, let's say, plasmid transfection, and we are ready to go. So for that reason, we at this stage decided to stay with HepG2 cells. But it's a fair question.
Yeah. Well, we're at the hour. We have quite a few more questions. I wonder maybe we can have another 5 minutes of your time.
I'm okay. Yeah, if attendees are willing to stick around, then
Okay.
I'm okay.
Let's see here. Okay, so to ToxTracker, does the potency in the assay correlate with carcinogenic potency?
Ooh.
That's a big question.
That's a good question, and in all honesty, I cannot really answer that question. So far, all of the obviously, if we look at how well does the assay identify genotoxic carcinogens, then it obviously is very good. The potency in dosing is very difficult. I think it's almost impossible to do that, in all honesty, because typically, of course, carcinogenicity is, first of all, it's a very complex disease, and typically is a result of long-term exposure and multiple changes in those cells. Where in ToxTracker, of course, it's an acute exposure, and you look at an immediate response. And there's a more complicating factor there. It's also a significant part of the cancers are caused by non-genotoxic compounds, so by non-genotoxic carcinogens. So I think where the BMD approaches to predict, let's say, genotoxicity in vivo works really well. Currently within the GTTC, so the Genetic Toxicology Expert Committee at HESI, there's ongoing activities to really use the BMD for ToxTracker to do the IV-IVE, so in vitro to in vivo extrapolation, including also PBPK modeling, and really looking at in vivo exposures, and that seemed to work really nice, but it's all related to gene tox and carcinogenicity. At some point, we will definitely try to do the comparison, but my prediction will be that it would be difficult, in all honesty.
There's a question about whether you've tested mixtures in either of the assays.
Yeah. For Tox Profiler, a limited number of mixtures have been tested. Obviously, you can only make a statement about the effects of those mixtures, but that has been done. More extensive work has been done in ToxTracker, in all honesty, where we've tested a whole range of different mixtures, being either very complex petrochemical products that contain I don't know, maybe 1 or 10 million different molecules, to environmental substances where pollutants in soil, for instance, or in groundwater was tested, but also artificial mixtures, extracts from packaging materials, extracts from let's say food extracts, and it typically works well in a sense that the assay is very sensitive, so you're able to identify, let's say, genotoxic pollutants or impurities or in those mixtures. If there's indication of what, let's say, pollutant is present in that mixture, you can of course try to spike those mixtures or make artificial mixtures to look for the, let's say, effect, and also understand, let's say, the different toxicology behind that mixture. Obviously, what the assay cannot do is say, so here we have a mixture and it contains this genotoxic compound. So but yeah, we've published, and also in collaboration with others, definitely have quite a lot of experience in testing mixtures there.
Okay, great. That's a hot topic these days. So.
Yeah, I think sensitivity is often an issue with those mixtures because often the let's say the impurities, pollutants are present in very low concentration, and can you pick up those let's say toxic substances there.
So I'm going to close with one last question, and all of you, thank you for your questions. If you didn't get an answer, we will follow up by email afterwards. So the question is, in the end of your closing slides, you talked about next generation risk assessment and adaptation versus adversity. Could you expand a little bit on this, or tell us what you're thinking?
Yeah. I mean, I strongly believe that with all the tools that are available, the ones that I showed you, but if of course we are not the only one that are interested in generating these new tools. I think we more and more get in the situation where you can use these smart models, in vitro models, using cell lines, organoids, organ on a chip approaches, to really get a good understanding, quantitative understanding of how molecules behave in a biological system. And it's that mode of action information and quantitative information that is essential there. And I hope that someday we will be able to basically move away from animal testing, because animals are definitely not always a good predictor for human effects, and to really come to a situation where we can rely on in vitro and maybe in silico to predict effects for humans. But you need that mode of action information and quantitative information from those models there. And I think all the approaches, so the AOP approaches, define let's say all the events that lead to an adverse outcome. The new models that provide that mode of action information, the quantitative dose response modeling approaches and software, and also the improvement of those in silico models together is, in my opinion, the way forward. And hopefully at some point we will be able to get away from most of the animal testing. There's a couple of areas where there will be difficult when we start talking about immune system and hormonal system. Developmental toxicity or embryo toxicity, embryonic development is obviously quite difficult to mimic in a petri dish. But I think we're making big progress, and yeah, I would only say the more people use it, the wider the acceptance, and the more support we get for these new approaches.
Yeah. Well, that's a great note to end our webinar on. Thank you again, Giel, for your presentation. Thanks all of you for, my dog's decided it's time to go out. Oh. Thanks for all of you for attending, and we'll see you next time. Take care.
Thank you very much, and have a nice day.
Thanks, you too.